Hello. So you guys are in lab if you're seeing this. So what we're doing right now is we're going to be going over part B of the test, so the short answer. And I'm going to be giving you some not so good examples of answers and some good examples of answers, because I think that's one thing that we have to work on. Also, handwriting. And this test, I've noticed, we really have to work on handwriting. Now, after I'm done going over these, the sub is going to go through the multiple choice with you and give you the answers to that. Now, listen, if you are not done with your tests, you should not be sitting through this video. What you should do is you should raise your hand. Okay? Now, if you have not started, then your substitute will give you a blank copy and you can work on it in the hallway. More than 2 people in the hallway, though, someone will have to go to the testing center. Okay? And if you have started your test and you haven't finished, then I have it right here, and I'm just going to show you where it is so you know where it is. Right here, this thing right here. So first period, everything in the folder. See how there's a folder underneath? Everything in the folder is stuff I need to hand back, and everything on top, tests that are not finished. Okay? So if you have not finished your test, you need to do that. I am not giving back the test yet because, well, obviously there are people who have started the test and haven't finished. If you don't do it by the end of today, I am going to come in tomorrow and grade them as is. Okay? And then, of course, you'll get a chance to retake, but don't rely on that, like I say every time. All right. So I thought this test was really good for you guys, actually. I saw some really good grades. All right. So let's talk about a few of these. The R, part B. I know you don't have a test in front of you, so you just have to do your best to read the screen and remember what it said. So we have the first question right here. Oh, that's backwards. I can't read that. Sorry. Okay, it says, base your answer to the questions in the diagram below and your knowledge of biology. State one possible reason why a gene for the production of human hormone would be placed into bacterial DNA. So this was a note, okay? I actually went over this with you on a video. So, this time you can't say that I never talked to you about it because I have video proof. All right, this is the segment of human DNA. This is the DNA we're going, kind of circular DNA we're going to put it into. We are cutting and copying and pasting, so that means enzymes are involved. And when you put it inside the DNA, put it inside the bacterial cell is going to do 2 things. First, it is going to make copies of that gene because all things that are single cellular reproduce using mitosis, all organisms that are single cellular. And mitosis means it will create exact copies. So when it creates exact copies of all the DNA that's inside, it's going to replicate that little piece of DNA from a human. Okay? You also know that genes, which are the little pieces of DNA, code for the production of what? Proteins. All right? So another thing that it will do is it will probably use the DNA and translate it into the production of a protein. So the question is, let me read it again. Why? Why would we put a gene that produces a human hormone inside bacterial DNA? So you can say a couple of different things. You can say we would put it inside bacterial DNA to make extra copies of the gene. Or you can say we would put it inside bacterial DNA so the DNA will create the protein that the gene codes for. So either one of those is good. There were variations A lot of people use the bat point on this, which is okay. Just make sure that you understand it now. Okay. 2. Humans have blue or brown eyes. Brown eyes are a dominant trait, while blue eyes are a recessive trait. Describe the offspring. Chalkboard. Describe the offspring when a brown-eyed hybrid person, big B, little b, mates with a blue-eyed person, little b, little b. Oops. Chalk. So, I'm going to draw my... I hate this board. Hold on. That doesn't help. Okay. Sorry. Nothing is helping. Can you see that? That's good. Okay. So, big B, little b, and little b, little b. We know that the big B codes for brown eyes because brown eyes are dominant. We know that the little b codes for blue eyes because blue eyes are recessive. Okay? So when we complete this is an awful piece of chalk. Oh, new chalk. Okay. I think it's a good kind. So when we cross this, which you guys are all experts at, right? Yes. We will get. Oh, it's a bad chalk. Okay, sorry. We will get big B, little b, little b, big B, little b, and little b. So the answer to this question, let's get to what they're actually asking. It says, describe the offspring when, these two people mate and create offspring. So if you were to describe them, you should probably talk about their phenotypes. So what they're showing, not just their genotypes. These are their genotypes, okay? But what are they going to show? Well, if this one has one dominant and one recessive, it's going to show the dominant because it only takes one dominant to show that trait, to overshadow the recessive. So, brown. 2 out of 4 will be brown. So 50% will be brown-eyed, which means that 50% will also be blue-eyed. So the correct answer is 50-50, or 50% will have brown eyes and 50% will have blue eyes. It's not enough just to draw the Punnett square. I'm glad that you did, if you did that. But have to also talk about what they look like. Okay, sorry about that. Next one. So, using the DNA sequences below, identify the 2 species that are more closely related and support your answer. So, number 3 right there. Those sequences are DNA codes. All right. So it's a DNA strand for A, DNA strand for B, DNA strand for C. So what you had to say, or what you had to look out for were, which 2 had the most similar DNA. And in this case, it would be A and B because they have the smallest amount of differences. Okay. If you have more DNA in common, it means that you are more closely related. So these are the DNA codes. That's all DNA. All right. So you will have more DNA in common with your mom than your aunt because you're more closely related to your mom than you are your aunt. All right. 4. All organisms receive inherited traits from their parents. Describe 2 inherited traits this tiger cub received from its parents. And then a lot of people were like, but I can't really see the tiger cub. That's okay. You don't have to. All you have to know is what kind of traits a tiger cub has. And really, you just had to say any 2 things about the tiger cub. Any inheritable traits. And I thought people did a really good job with that question. I saw a lot of stripes and orange fur and rounded ears and sharp teeth. It's all great. Good job. Okay. Because remember, we receive all of our traits from our parents. Okay, 5. So, I'm going to read it, then I'll show you. It says, caretakers at a zoo are trying to determine which of the 2 male tigers fathered the newest cub. They obtained DNA from the tiger cub, the mother tiger, and 2 male tigers. The DNA was analyzed. The results of the analysis are shown below. What process is that, guys? Gel electrophoresis, right. That's gel electrophoresis. When you break up the DNA using restriction enzymes and you put them up at wells at the top, and then you create a positive charge down here, and the DNA is pulled across the gel because DNA is negative, and they create a positive charge down here. And they're going to be pulled at different rates because they're different sizes. So the bits down here are going to be smaller because if they have less mass, they can travel farther in the same amount of time. And the bits up here are going to be longer. All right. Anyway, basically it's just a who's the daddy type of thing. And I got a lot of answers that I accepted. All you had to do was this. Is that going to work? Okay, we're going to make this work. So here's your cub right here. And here's mom. You had to do, this is the easiest way to do it. And you guys did it several different ways. You could have said, okay, cub has this gene with mom. And then it has this gene with mom and this gene with mom. So that means a pile of DNA. So that means that this piece of DNA must have come from dad. So if you were to follow it across, it would go past male 2, okay? And it would go all the way over to male 1, okay? So male 1 is the father, okay? See how I did that? All right. It's wrong to say that male 1 is the father because he has more DNA in common, because that's not really, I mean, I gave you guys the credit for that, but keep in mind, that's not really what we're looking for. We're not looking for having the most DNA in common with the cub. We're looking for, the cub's DNA, the DNA that didn't come from mom had to have come from dad, right? So the DNA that wasn't from mom had to come from male 1, not male 2. Okay? No. All right. Next question. So I'll just cut to the chase right here. The reason why I put this on the test is because it's a great example of sometimes when the Regents gives you these big, long questions with a ton of information that you don't really need. And then they ask you a question at the end that, it has a lot to do with the text, obviously, but you probably could have answered without reading the text. So that question is, explain how cancer cells differ from normal cells. And I got a lot of great answers for that. A lot of, I wonder why, right? A lot of, cancer cells have an abnormal rate of growth. Cancer cells grow too much. Cancer cells can harm you. That's still good. Cancer cells have abnormal mitotic division. That's all good. Great. All right. 7. So I backed it up with this one because this one is a question that was on the Regents that you have to read. So I can't read like this, so I'm just going to turn it around and read to you. It's about grasshopper mice. So in the Sonoran Desert in the southwestern United States, the grasshopper mouse is active at night, scratching for crickets, rodents, tarantulas, and even scorpions. The mouse ignores the venom of the scorpion, kills it, and consumes its flesh. The ability of the mouse to ignore the pain normally associated with the venom of the scorpion is due to the presence of a mutated protein. This protein prevents the pain signal from reaching the brain. These mice are born killers, capable of taking down prey that are much larger than themselves. They are also aggressive neighbors and take over nests by displacing other desert inhabitants rather than making their own. Under difficult environmental conditions, they may even eat members of their own species. Nice guy. All right, the question is, explain how research on the mutated protein identified in the grasshopper mouse could benefit humans suffering from chronic pain. Really simply here, I mean, some of you guys gave great in-depth answers. A really simple way to answer this would just be it could benefit humans because because that protein allows the mouse not to feel pain. So we could figure out a way, take that gene, put it in human DNA, and then humans wouldn't have to feel pain, chronic pain. All right. So we could figure out a way to incorporate that mutated gene into our own DNA. Okay. And no, it's not something we can do right now, but your answers for biology questions like this don't have to be stuff that we already know how to do. You can kind of go out on a limb and say, well, you can figure out how to do this. That's what makes you a scientist, right? Figuring things out. All right. Coding. This lovely chart. So I think for this, people either knew exactly what they were doing or they literally had no idea what they were doing. All you had to do is you just had to figure out the RNA code and then get the amino acid sequence by using this chart right here. So we'll try this again. Does that work? Can you see it? Okay. All right. So if you are going from DNA to RNA, then instead of A's going to T's, they're going to go to U's. C's will go to G's, G's will go to C's. And then when you have a T, it will go to A. So that means this will be U, G, C. Okay, that's your RNA. And then up here it says CGC, so that will be G, C, G. And then ACC will be U, G, G. You guys saying this with me? And then this one says TTA, so it will be, oh, brain fart, A, U. Okay? Now keep in mind, when you use this chart up here, you can't use your DNA. And I saw a lot of you stuck on that and I couldn't help you. You can't use the DNA. You have to use the mRNA, which is what you just translated, what I just translated. Hopefully you did it too. All right, so I take my translation, UGC, and I go, can you see that? Oh, yeah. I go first base U, second base G, second base G. So it's in the square here. UGC, CYS. Okay. And then GCG, first base G, second base C, follow it down, GCGALA. So cis, ALA. Okay, we'll do the last two. UGG, right there, TRP. Oop. And then AAU, ASN. All right. So that is it for part B. If you want to retake this test, I would recommend doing it as soon as you can, because remember, the makeup test is just this test rearranged a little bit, but it's still, you'll still forget it. I just reminded you of everything you need to know on part B. You will get part A from the sub and study it and come back and take it. Okay? And if you have any questions for me on anything, make sure to let me know through the Remind app. Don't forget to turn in your relationships and biodiversity lab. I am missing a lot of those. Okay? A lot. You don't want to be the person with 5 missing labs on lab check day, which is Thursday and Friday. Okay? All right. Bye.